Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh. A very cheerful day to Dr. Nurjahan and my fellow classmates. Today we are from Group 1 who would like to present about future cell and gene therapy for osteoarthritis using mammalian protein production platform. Before we continue to the presentation further, I would like to introduce myself, Imanina Nazifa, and my partner for this presentation, Aimi Diana. Our group members are Nur Farah Izzati binti Jumsi, Yu Yi Ying, Husna Izian binti Ishaq, Prima Lata, Pierre, Siti Nur Suryani, and Tri Bangladesh. The presentation today will be divided into several parts which you can see from this table of content. Hope you guys will enjoy the presentation.
Hi, I am Aimi. So firstly, I want to introduce about the osteoarthritis. Osteoarthritis, or OE for short, is a type of joint disorder with degenerative and inflammatory components. According to research, this disease increases worldwide and women are severely disabled by this disease compared to men. Currently, there is no cure for OE, but there are several steps to prevent it, like maintain healthy weight and regular exercise, surgery, and so on. OA tends to reduce the mobility of a person, which can eventually result in some other diseases, such as cardiovascular disease, diabetes, hypertension, depression, and anxiety. That is also the result of OA due to the sleep disturbance. So as you know, symptoms of OE are joint pain, stiffness, tenderness, loss of flexibility, crepitus and grating sensation, joint effusion, and swelling. So fortunately, there are several steps to prevent OA, which are maintaining healthy weight and regular exercise, intake of anti-inflammatory medication, surgery, and joint arthroplasty.
Next, we will move on to the hallmark of osteoarthritis. Usually, all major diseases are characterized by hallmarks, which are the key biological alterations that occur in a disease. For example, research stated that cancer comprises 6 biological capabilities that are gradually acquired during the multi-step development of human tumors. The hallmarks of aging occur in osteoarthritis. So nowadays, the world population has been continuously increasing and progressively aging. Aging is characterized by a complex and intra-individual process associated with 9 major cellular and molecular hallmarks, which are genomic instability, telomere attrition, epigenetic alteration, loss of proteostasis, deregulated nutrient sensing, mitochondrial dysfunction, cellular senescence, stem cell exhaustion, and altered intercellular communication. So the major hallmark of OA can be observed from the gradual loss and degradation of articular cartilage. The cartilage thickness used to determine the efficacy of new regenerative treatments. Next, cartilage degrades are caused by excess weight and the pro-inflammatory cytokines, which is adipokines, and leptin produced by white adipose tissue in obese and overweight OA patients.
Okay, after understanding the hallmark of osteoarthritis, we will now look at the molecular alteration of cartilage. Biochemical, inflammatory, and metabolic factors are key roles in the initiation and progression of OA through alteration of chondral, subchondral, and synovial membrane. And the presence of pro-catabolic and pro-inflammatory factors can cause activation and inflammation of the chondrocyte, which lead to the phenotypic shift through a phenomenon called chondrosenescence and the development of a senescence-associated secretory phenotype. It also may cause formation of phenomenon that disrupt the homeostasis and metabolism in the cartilage, the change in the cellular metabolism in the chondrocytes, and inflammation of synovial membrane, which will lead to the synovitis. The drug for OA is only reducing the symptom and inflammatory such as inhibit matrix degrading enzyme and bring cartilage metabolism back into the equilibrium. Today, all the scientists have focused on the improvement of anabolic activity for structure modification instead of inhibiting catabolic activity for symptom modification. However, native and primary cells are unlikely to have capacity for producing sufficient quantity for growth factors for stimulating cartilage repair.
Now, let us move on to mammalian protein production platforms, a potential technology to cure OA. So, mammalian protein production platforms are indispensable cell factories that are used for large-scale production of antibodies and therapeutic proteins. Expression of antibodies and mammalian cells is key technology that is important to research about functional studies on human and higher eukaryotic genes. Expression systems allow production of proteins, especially those of clinical relevance and human origin. So, recombinant protein and a vast array of antibodies are now produced in mammalian cell lines instead of bacterial expression systems. This is to ensure proper protein folding and post-translational modification essential for full biological activity, properly introduced in a eukaryotic and mammalian context. And mammalian cell expression system are one of the dominant tools for producing complex biotherapeutic proteins. The mammalian cell lines that are commonly used in the research and industrial therapeutic protein production settings are Chinese hamster ovary cells and human embryonic kidney 293 cells. So nowadays, various mammalian and non-mammalian expression systems are being used for protein and glycoprotein production. Cellular engineering strategies have been developed to increase protein and glycoprotein productivity. One of the most technologies that is successfully being developed is called omics technology. Omics technologies are continually being used to improve cellular expression system and enhance such platforms for therapeutic protein production. Next, the method for producing large quantities of antibodies are done by transient expression systems in mammalian cells by using a combination of simian virus 40, SV40, large T antigen P21 and P27. It has been possible to scale up expression level of antibodies from 0.1 ml up to 1,200 milliliter in bioreactors. So the biotechnology companies can produce sufficient quantities of therapeutic antibodies and protein for tests in preclinical studies, and also for early phase clinical trials through the ability to scale up.
Okay, so next, let us dive into our platforms for overproduction of recombinant growth factors. Access to mammalian cells models that truly mimic chondrocytes with phenotypic chondrocytic properties can be useful for pharmaceutical or biotech companies that want to make large quantities of growth factor capable of stimulating cartilage matrix synthesis. At present, there are no cell lines derived from cartilage or other joints tissue that are truly comparable to CHO or HEK293 cells. So, cellular models must be employed, including CHO, HEK293 cells, and their derivatives such as GP2293 is immortalized cell lines that function as cellular factories for overproduction of proteins. Then, GP2293 cells are specialized protein packaging cells that function as specialized transfection models, protein packaging tools, and cellular factories. GP2293 cells are commonly used for overproduction of target human proteins. It is also a promising candidate for overproducing therapeutic proteins and growth factors that native primary cells cannot produce in sufficiently large quantities, either short or long term. Also, the cells can be irradiated to obliterate their proliferation capacity in remaining protein packaging cellular factories, but without the ability to proliferate. The cells are feasible in cellular therapies for OA through elimination of proliferation through irradiation, especially if the cells are to be injected into the closed microenvironment of the synovial joint, isolated from the circulatory system. Irradiated cells retain their capacity from protein overproduction, but they cannot divide and proliferate, which means that they will die shortly after being injected into the joint.
So, after understanding the theoretical part, let us look at some real-life examples in case study. The case study will be focusing on production of transforming growth factor beta-1, TGF-beta-1 by GP2-293 in the colon tissue gene cell and gene therapy product TGC. So, TGC is a unique first-in-class cell and gene therapy targeting NE-OA through a single intra-articular injection of joint-derived chondrocytes, irradiated GP2-293 cells. In the future possibility in the treatment of osteoarthritis due to their overproduction of biological growth factors, which promote anabolic repair and regeneration, expected to recruit over 1,000 patients for clinical trials to assess the pain and physical function endpoints, imaging outcomes, radiograph, and wet biochemical markers in patients receiving TGC treatment. The clinical trials have shown some degree of pain relief, increased mobility, as well as indications of reduced osteoarthritis progressions and improvements in joint structure, function, and reduced stiffness. Other than that, allogenic drugs containing cells and biological growth factors could be an alternative to traditional pharmacological osteoarthritis treatments, which tend to provide unintended side effects. The ultimate aim is to achieve symptom and structure modification till the measure that the knee joint is preserved and surgery is delayed for a sustained amount of time.
So next, let me talk about the DMOAD status from TGC. Colon Tissue Gene conducted a phase 2 clinical trial which showed a 2-year improvement of pain and function. These efforts were sought to be continued by running a national phase 3 clinical trial. Colon Tissue Gene has architectured a trial to seek a disease-modifying osteoarthritis drug, which is DMOAD, designation for TGC from the FDA.
Next, let us look at the use of GP2-293 cells in TGC. Firstly, the human GP2-293 cell line is one of the most key components of TGC. These cells carry out the vital function of overproducing the crucially important growth factor TGF-beta-1. And then GP2-293 cells have been used throughout the development process from the first production of the master cells bank, MCB, and the next step is the development of the working cell bank and the final product formulation. GP2-293 is a HEK293-based retroviral packaging cell line used for a large-scale protein production. It is being used as cellular platform for overproduction of therapeutically relevant human proteins. It is also the first employment of a human protein production platform for osteoarthritis treatment and cartilage regeneration.
Okay, so finally, we move on to the last topic of the safety of GP2-293 cells in TGC. Transduced and irradiated Gp2-293 cells cannot proliferate. Thus, the eGP2-293 cells in PDC cannot survive and proliferate into the joint. Therefore, the cells cannot survive for more than a very short period after being injected into the joint. Korean Food and Drug Administration has stated that in VOSA, the Korean version of TGC is safe as cells don't survive outside 44 days. After the cells TGF-beta1 overproduction duties, they will die and their remains will be cleared by joint resident inflammatory macrophage by phagocytosis. The scientific basis for the use of mammalian cell transfection models is clear in the development of the TGC. The efficacy and safety of HEK293 cells and their derivatives in regenerative medicine has not been truly researched, but the prospect for future use of transfection tools in regenerative medicine and cell therapy has a vast amount of opportunity. Okay, so before we end our presentation, let us summarize some of the key points from today's presentation. The progressive loss and deterioration of articular cartilage is a major feature of OA. Drug for OA is only reducing the symptom and inflammatory. Today, all the scientists have focused on the improvement of anabolic activity for structure modification instead of inhibiting catabolic activity for symptom modification. However, native and primary cells are unlikely to have capacity for producing sufficient quantity for growth factors and for stimulating cartilage repair. There are no cell lines produced from cartilage or other joint tissues that are genuinely equivalent to the CHO or HEK293 cells at the moment. As a result, cellular models such as CHO, HEK293 cells and their derivatives such as GP2-293 are used. CHO, HEK293 cells and their derivatives such as GP2-293 are immortalized cell lines that work as cellular factories for protein overproduction. The clinical trials have shown some degree of pain relief, increased mobility as well as indication of reduced OA progressions and improvement in joint structure, function and reduced stiffness by implementing production of transforming growth factor beta-1, which is TGF-beta1 by GP2-293 in the colon tissue gene cells and gene therapy product. So we believe that in the future, with intensive research and clinical trials, scientists are able to find a cure for OA and end the suffering of all generations. So I think that's all from us. Hope you guys learned something from this presentation and thank you for listening.